I'm a clinician who takes care of patients who have genetic disorders, and sometimes we can't figure out what the genetic cause of their disease is. And I know that you're a principal investigator in the Duke Center for Combinatorial Gene Regulation, which is an NIH Center for Excellence in Genomic Sciences. Can you talk a little bit about the work that you've been doing with your CEGS grant and what that kind of looks like for the face of how we solve these clinical problems?
Yeah, so that's been a super interesting project. It's been really amazing. It brings together 7 different PIs from Duke. It includes folks that are purely clinical in their research. It involves folks that are purely computational and statistical. It involves people that have only ever worked on cell lines in the lab, and it really brings all these people together in an effort to try to, as you say, connect the dots between some of these clinical diseases and the genomes and the underlying causes. So the CEGS itself is taken on a hypothesis that one of the reasons we're not being able to diagnose these patients is because we're only looking at one variant at a time. We might say, okay, it's this one variant, the other variant. Whereas we think there's a huge amount of missing information by not looking at how combinations of variants might act together. And those combinations can happen at different scales. It could be they have 2 genetic variants right next to each other that are somehow contributing to disease more than you might expect just from one. It might be that they have 2 variants that are very far apart, but the genes that they impact are themselves interacting with each other and causing greater effects, and so on and so forth. And so we've been really pushing hard both to develop new technologies to allow us to understand how these combinations of variants act together, and then very clearly keeping that close to the clinic. We don't want to just develop these technologies and have them kind of fly off into space. We want to make sure that we're bringing them back to clinical cases on a regular basis. And I mean, to some of the point you raised, we're working really hard on those clinical cases to focus on the patients that have really eluded genetic diagnosis so far, right? Patients that have been through the gamut of phenotyping and genetic testing and, in some cases even whole genome sequencing, and it's not really clear what's causing the disease that they may have, right? We've had some interesting successes early on. We've already found, I think, 3 or 4 patients where we've been able to pretty definitively define what is the actual cause of their disease, identify new causes of these rare diseases. But I think one of the things we've learned along the way, right, is that every rare disease is special in its own way. And so one of the hard things that we've been really struggling with is, can we start to get some common themes? Can we start to put some principles in place that will allow us to make this much more routine? We've actually, much to our surprise, we went in with a hypothesis that there was going to be a lot of gene regulatory variation that we were finding. What we keep finding time and time again is splicing related variation, not something that we really intended to find, but it's also something that doesn't get really looked for nearly as much as you might expect with normal genetic testing. And so sort of spinning out of that CEGS project, there's been a sort of a whole new emphasis of our team on understanding how indeed changes in splicing are contributing to rare disease in ways that haven't been identified before. What's exciting about that is, when it comes to a gene regulatory element having a genetic variant, sometimes it's a little hard to think about how you might benefit that patient. But when we coming across these gene splicing related variants, there's already in place a whole suite of molecules that can impact splicing for genes in many different ways. And so there's often a really natural path to start thinking about, how you could even develop, clinical care based on this, right? It's always challenging when these cases are rare. You can't do clinical trials like you're normally thinking about it. But that's not stopping us from pushing ahead and at least trying to, develop the approaches and the analyses and the methods to get these molecules as far as we can, so that way, if indeed they can help these patients, we're ready.
That's so incredible, right? I mean, 2 really important themes. One is for a given patient and their family members to understand what the risk genetics might be, but also importantly, incredible work where you might actually be able to develop therapeutic advances. And this is a, very important topic as we think about therapies in these rare and orphan diseases. So really, really incredible work.